Alright, in this recording, what we want to start to think about is what happens to a protein after translation is finished. And this is a huge topic of post-translational targeting and modification. Many proteins need further work done to them before they're functional or in the right place in the cell to do their job. So, though we understand translation, there's a whole series, a plethora of different systems that are involved in then taking that protein and modifying it and targeting it. So we want to understand a basic idea of how that works. Now, some polypeptide chains will fold spontaneously into the three-dimensional shape in the cytoplasm, ready to do their job. Some need other proteins to work on them to actually cut them down, and others need to assemble into larger complexes to work. So this is just a few examples, and we'll talk about more as we talk about control of proteins and ultimately gene expression. Now, one of the things to be concerned about is the location of a protein. Especially in a eukaryotic cell, you've got many options. Are you headed for the cytoplasm? Are you going back to the nucleus? Mitochondria, chloroplast, where in those organelles? Are you headed for the plasma membrane, right, to be secreted? And there's many mechanisms to get to each of those locations. But we remember that the eukaryotic cell is like a mansion. How do you know where in that mansion, what room in that mansion are you supposed to do your job? We have the additional room of the central vacuole and the chloroplast in plant cells as well that we want to think about. So what we talk about is protein targeting. And we talk about a targeting peptide. So but before we talk about that structure of the protein and how it knows where to go, we have to go back to the place where proteins are synthesized. So the location of protein synthesis is on the ribosome. And now we get to use some of the information that we learned earlier, and that is the anatomy of the cell and the endomembrane system. And the ribosomes are in one of two places in a eukaryotic cell. They're either free in the cytoplasm or they're attached to the ER, making it rough ER. Remember, rough ER is rough because it's studded with all these ribosomes. Well, in fact, those ribosomes are all one big population. And the ribosomes that are actually on the ER are there because they're going through a process called Cotranslational Translocation. So co with translation, translocation, moving to a different spot. So all ribosomes are going to engage the mRNA free in the cytoplasm. Some of those will translate the protein and let it go free in the cytoplasm. Others will begin translation and then pause because there's the amino acids, the initial amino acids that emerge from the ribosome actually are what are called a signal peptide. And that signal peptide will bind to another protein called a signal recognition particle, the SRP. That SRP halts translation and basically targets the entire ribosome mRNA complex to the ER, where it binds, and then the protein synthesis can continue, but instead of releasing the protein free to the cytoplasm, what in fact it's going to do is, as it exits the ribosome, it's directly fed across a pore, which is made out of a protein, into the ER. So remember, those proteins that are going to need glycosylation, the addition of sugars, are targeted to the ER, the beginning of the endomembrane system, the beginning of that pathway that protein could take. And so that signal recognition particle has to be encoded for in the gene to ultimately get that protein to be glycosylated so that it can function properly. So all this information can be found in the DNA, but then functionally lots of pieces of machinery have to come into play to get that protein to work. Now, again, we're always going to start with our ribosomes in the cytoplasm, right? And so whether or not they're targeted by the SRP to the ER or they translate that protein free in the cytoplasm depends on the protein. So what about those proteins, though, that are translated free in the cytoplasm? Do they all just reside in the cytoplasm? No. So many of those proteins may be targeted by, again, a targeting peptide to either the nucleus or mitochondria or chloroplasts or peroxisomes, or potentially even to the ER post-translationally. So there has to be some information, again, in that newly translated peptide that targets the protein. And so what we're going to see is that information can be in many different locations. So that targeting peptide can also be known as a signal sequence on the N-terminus, which ultimately will be cleaved, cut off by another protein, a protease. We may see other locations of that information. It can be internally localized, or you can see it on the C-terminus. So the language here, again, signal sequence, targeting peptide, transit peptide, all these have slightly different connotations, but you can pretty much use them interchangeably. It's generally 20 to 40 amino acids at the beginning of that peptide. But again, you can also find targeting information internal to the protein or on the C-terminus. So again, this is just to introduce to you the idea that, once a protein is translated, there are many, many other things that have to come into play before it can really do its job properly in many cases.